Hello students, I am Dr. Nidhi Agarwal. Welcome to EPG Pathshala. Today we will study about phase 1, 2 and 3 of clinical trials under the module Product Development Part 2. In this we will learn about different phases of clinical trials, their designing and objectives. Coming to the definition of clinical trial. Clinical trial means a systematic study of new drugs in human subjects to generate data for discovering and or verifying the clinical, pharmacological, including pharmacodynamic and pharmacokinetic effects and adverse effect with the objective of determining the safety and efficacy of the new drug. Coming to clinical trials and their purposes. They are basically being conducted to move a drug through a development program, to gain marketing authorization, to guide treatment of individual patients, to investigate a specific property of the drug such as incidence of an adverse event, and lastly to select one drug rather than another for addition to a therapeutic formulary and inform a health policy. Coming to the phases of clinical trial. Clinical trials are being conducted in different phases. Phase 1, which is said to be human pharmacology. Phase 2, which is therapeutic exploratory trials. Phase 3, therapeutic confirmatory trials. Phase 4, post-marketing surveillance. Coming to the first one, phase 1 clinical trials, which are said to be the human pharmacology trials. They are conducted in healthy volunteers, preferably be carried out by the investigators trained in clinical pharmacology with access to the necessary facilities to closely observe and monitor the subjects. It includes 2,200 subjects. Drugs with significant potential toxicity, example, cytotoxic drugs, and the drugs which are required to treat the HIV patients are usually studied in patients themselves. In the phase 1 trial, doses are increased if there are no severe side effects and patients are tested to determine if he or she is responding to the therapy. The dose escalation studies are used to determine the best and safest route that can be administered and is a fraction of the dose that cause harm during animal testing. Testing is usually sequential, with data being reviewed after every patient or small group of patients. Dose toxicity and dose efficacy curves are determined during this phase. Phase 1 clinical trial include single ascending dose trial, that is termed as phase 1A, and multiple ascending dose trial, that is phase 1B, and the food effect studies. Coming to the objectives of phase 1 trial. Estimation of safety and tolerability with the initial administration of an investigational new drug into human is being done in phase 1 trial. The maximum tolerated dose is being determined in order to see the expected dose range which will be required for the later clinical studies. It is also required to determine the nature of adverse reactions that can be expected. It may include single or multiple dose administration. The pharmacokinetic profile is also being assessed in the subjects, which particularly emphasizes on the absorption, distribution, metabolism and excretion pattern of the drug. The pharmacodynamic studies may also be done in healthy volunteer subjects or in patients with the target disease. They are basically done for the early measurement of drug activity. They are generally performed in later phases but may be appropriate when drug activity is readily available. Examples of phase 1 trials are: first, dose tolerability trials; second, single and multiple dose pharmacokinetic or pharmacodynamic trials, trials in some special group of patients, and drug-drug interaction. Coming to the next phase of clinical trial, that is phase 2 clinical trial. They are termed to be therapeutic exploratory trials. The phase 2 clinical trials are conducted in a group of patients and they should be closely monitored. Doses used in phase 2 clinical trials are usually, but not always, less than the highest doses used in phase 1 trials. These studies are performed on larger group comprises of 100 to 300 subjects and are designed to assess how well the drug works and to continue safety assessment. Therapeutic doses which are determined during phase 1 are administered in the phase 2 trials and the patients are monitored closely by the investigator. The phase 2 trials are often conducted in multi-institution setting. The phase 2 trial can further be divided into phase 2A and phase 2B. Phase 2A trials are the pilot clinical trials required to evaluate the efficacy and safety in selected populations with the disease or condition to be treated, diagnosed or prevented. Objectives may be on dose response, type of patient, frequency of dosing, or other identifiers of safety and efficacy. Coming to phase 2B trials, these are most rigorous trials designed to demonstrate efficacy. The development process usually fails during this phase 2 trial when the drug is discovered not to work as a planned or to have toxic effect. The phase 2 design depends on the quality and adequacy of phase 1 studies. The single stage and multiple stage phase 2 clinical trial designs are often developed on the basis that one endpoint is of interest. Initial therapeutic exploratory studies may use a variety of study designs, including concurrent controls and comparison with the baseline status. Subsequent trials are usually randomized and concurrently controlled to evaluate the efficacy of the drug and its safety for a particular therapeutic indication. Coming to the objectives of phase 2 trial. The phase 2 trial are done to evaluate the effectiveness of a drug for a particular indication or indications in patients with the condition under study. Phase 2 trials are designed to determine the common short-term side effects and risks associated with the drug. Phase 2 trials are also done in order to evaluate the potential study endpoints, therapeutic regimen, which includes the concomitant medication in addition to the interventional drug, and target population, example, mild versus severe disease, for further studies in phase 2 or phase 3. Coming to the other features of phase 2 trial, they are the earliest trial of short duration in narrowly defined subject population, which comprises of patients with the disease of interest. They are done using certain biomarkers or the surrogate endpoints in order to assess the efficacy and safety of the drug. Now coming to the important phase 3 clinical trials. They are often termed as therapeutic confirmatory trials. These trials are the most rigorous and extensive type of scientific clinical investigation of a new treatment. These are also termed as the pre-marketing phase of clinical trials. These trials are usually most expensive and time consuming. These trials may be difficult to design and run. They involve a large number of subjects, which could be between 100 and 3,000 subjects. They are being recruited at different trial sites. Coming to the designs used to conduct a phase 3 trial. The phase 3 trial should be randomized control trial. It should either have a parallel design or it can be an uncontrolled trial where a single treatment can be used. Other designs which are used for the phase 3 trials include no randomized concurrent trials, factorial designs, and group sequential designs. In the phase 3 trials, patients are monitored by the investigator as well as the physician. The phase 3 trials may be divided into two, phase 3A and phase 3B. Coming to phase 3A trial, these are the trials which are done after efficacy of the therapy is demonstrated, but before the regulatory submission of a new drug application or other dossier. Phase 3B trials, which are conducted after submission of a new drug application or other dossier, but before approval and launch of the drug. The phase 3 clinical trials have been designated to be the gold standard. Coming to the objectives of phase 3 clinical trials. They are basically performed in order to demonstrate or confirm the therapeutic benefits of a new drug. These trials confirm the preliminary evidence accumulated in phase 2 trials that a drug is safe and effective for use in the intended indication and recipient population. The phase 3 trials are done to explore the dose-response relationships, that is, relationships among dose, drug concentration in blood and clinical response These are also done to test the use of drug in wider population in different stages of disease, or to check the safety and efficacy of the drug in combination with other drugs. The phase 3 clinical trials, they are large controlled trials to establish the efficacy of the drug. Examples of the design of phase 3 trials are randomized parallel dose response trials, clinical safety trials, trials of mortality or morbidity outcomes, large simple trials, and comparative trials. After the completion of phase 3 clinical trial, the sponsor composes a whole document which is now termed as a new drug application document, which he submits to the regulatory agency in order to get approval of the drug to be introduced into the market. The application for NDA or the new drug application should be submitted to the desired regulatory agency for the permission to market a new drug in the desired regulatory region. The data includes the whole non-clinical as well as the clinical data. There are three forms of new drug application. The three types of new drug application are full new drug application, paper new drug application, and abbreviated new drug application. Coming to the first one, full new drug application. For non-human or preclinical studies, no IND is required. To obtain chemical information necessary to set the exact specification for the active ingredient or to obtain the stability data, in vitro testing is being done. Animal toxicology tests are also done as per the good laboratory practices, and the accumulated data is the part of full NDA. The relevant information which is required for the submission of the investigational new drug application for filing further full NDA includes the safety and effectiveness of the new drug, the chemistry, manufacturing control information, pharmacology and toxicology information, previous human experience, and other pertinent information. The FDA evaluates this investigational new drug application for 30 days, and in the end, the FDA may approve, may pose a clinical hold, or may raise certain questions which need to be answered by the sponsor. And after the expiry of initial 30 days, IND can be amended. Now coming to the application to market a new drug for human use or as an antibiotic for human use. The new drug application must contain the following: index, summary, chemistry, manufacturing and control information, non-clinical pharmacology and toxicology data, human pharmacokinetics and bioavailability profile, microbiology data, clinical data, statistical analysis data, and the proposed labeling. Typical new drug application dossier consists of tens of thousands or even hundreds of thousands of papers. Coming to the classification of new drug application. New drug applications are classified by Food and Drug Administration with a code that reflects both the type of drug being submitted, and its intended use, which are as follows. First, a new molecular entity. Second, new salt of previously approved drug, which is not a new molecular entity. Third, new drug formulation of previously approved drug, not a new salt or a new molecular entity. Fourth, new combination of two or more drugs. Fifth, already marketed drug product or duplication. New indication for already marketed drug, and lastly, already marketed drug product with no previous approved new drug application. The Food and Drug Administration will review the document. The priority review is described by the following letter codes. First being the letter S, which indicate the standard review. Second, P, which indicates the priority review for drugs offering advantage over the existing drug. In the end of FDA review, three possible letters may be sent to the sponsor: not approvable letter, approvable letter, or the approval letter. In the first two cases, there may be certain concerns with the drugs which is to be launched. The queries raised by the FDA may be answered by the sponsor. Coming to the post-approval requirement. Following approval of a new drug application, FDA requires the submission of three types of reports by the sponsor. First, serious and unexpected adverse drug experiences. Second, periodic information on all adverse drug experiences as well as other safety and effectiveness profile. Third, information relating to all other aspects of the drug must be reported immediately to Food and Drug Administration if they represent a potential problem. The whole data also comprises of foreign as well as the domestic adverse experiences which are observed, and the other information should also be the part of these reports. Coming to user fees. Food and Drug Administration has authority to collect user fees for pioneering drug until generic competition is approved, and the fees include a one-time new drug application fee, an annual product fee, and an annual establishment fee. In return for receiving the user fee, the Food and Drug Administration has to commit to specific goals. Those are particularly related to the improvement of the review process of the dossier being filed. The FDA should also meet the specific timelines. The FDA should make improvement in the review process as well as in the approval process. It is the responsibility of the FDA to reduce the backlog of the applications filed to it. Coming to the second type of new drug application, that is the paper new drug application or section 505(b)(2) new drug application. It basically applies to those situations where a pioneer drug was unable to be protected by the patents or the market exclusivity, but where an applicant is unable to submit an abbreviated new drug application because the modified drug is somewhat differ from the pioneer drug. The paper NDA basically relies upon the pioneer NDA for all the information except the data needed to support the element of a substantial difference. The paper NDA need not include any data relating to the basic safety and effectiveness of the drug. Rather, it should include the difference between the pioneer drug and the modifications of that drug if there is any change in the safety or effectiveness. Coming to the third type of new drug application, that is abbreviated new drug application, which is often abbreviated as ANDA. An abbreviated new drug application may be approved by FDA for a generic version of a pioneer new drug after all the relevant product and use patents have expired for the pioneer drug and all relevant periods of the market exclusivity for the launched drug or the pioneer drug have also expired. Why these applications are called abbreviated? These applications are called abbreviated because generally they do not require to include the animal or the clinical data to establish the safety and effectiveness. However, the applicant or the sponsor need to demonstrate scientifically that their product is bioequivalent in terms of their pharmacokinetic profile. In what condition an ANDA can be submitted? The ANDA can be submitted if the generic version is the same as the pioneer version with respect to the excipient used to prepare their drug formulation. The generic version is different from the pioneer version in any significant respect, that is, a different active ingredient, a different route of administration, or a different dosage form with a different strength. The reviewers have a special focus in analyzing the submitted chemistry and microbiology data, bioequivalent data, the labeling information provided by the sponsor, as well as the request for the inspection of the plant where the drug will be manufactured. And if there is one or more essential components are missing from the filed application, then a refuse to file letter may be issued. Therefore, in the present module, we have covered aims and objectives and conduct of phase 1, 2 and 3 clinical trials. We have also learned the information regarding new drug application and its different types. An insight of abbreviated new drug application for generic drugs is also studied. A thorough information regarding filing a new drug application and abbreviated new drug application has also been covered. After the approval of new drug application, the drug is being marketed, which is followed by an important phase of clinical trial, which is also termed as post-marketing surveillance or the phase 4 trials. Thank you.